Good afternoon, everyone. My name is Molly from the Macromolecule Factory. Let me introduce y'all to where I work. Through this quick trip, you'll all get to know what our factory makes and how it affects the human body we work for. We work inside the human's digestive tract, to be specific. We meet a lot of other products occasionally floating outside our windows. There are a lot of other factories around us that have their own specialized jobs too. Come on, let's go!
Welcome to the Carbohydrate Station. In here, we are assembling atoms like carbon, hydrogen, and oxygen into monosaccharides, also known as simple sugars.
White is hydrogen, red is oxygen, black is carbon, and the gray links are bonds.
The most important monosaccharide we make is glucose. This one here is going to be an alpha glucose.
Half of these become beta glucose. They are different because of the position of the hydroxide bond to carbon 1. This is done by joining two monosaccharides through dehydration synthesis. In dehydration synthesis, a molecule of water forms as a product and the glycosidic linkages form between the two molecules. One gummy bear equals one monosaccharide.
There goes a monosaccharide.
There goes a disaccharide. Here goes polysaccharides, which are polymers with a few hundred to a few thousand monomers joined by glycosidic linkages. So basically, think of it as hundreds and thousands of disaccharides or monosaccharides. Oh my! It's bread! Full of starch molecules!
If we look closer, this is how the starches look like. Starch is a polymer of alpha-glucose monomers as granules within cellular structures known as plastids, which include chloroplasts. Usually a 1,4 linkage forms, meaning that if you number the carbon atoms, the first one of the first molecule and the fourth one of the second molecule are bonded together. Humans can't produce starches. However, humans have enzymes that can hydrolyze plant starch, making glucose. Some examples of starches in the human diet are rice, corn, and potato, as well as grains in general.
Oh no, not again! Here comes some cellulose in the form of salad. My human must be eating a lot right now. Cellulose is another thing that humans cannot make on their own. If you look closely, cellulose consists of only beta-glucose joined by glycosidic linkages, every other glucose monomer upside down relative to its neighbors.
Some animals have enzymes to digest cellulose, like cows, but for humans, cellulose helps with the smooth passage of food through the tract.
Warning, insufficient glycogen.
Uh-oh! Why isn't anything coming through the belt? Our human must not have enough glycogen. She must have went on a diet.
In humans, glycogen stores are depleted in about a day unless they are replenished by consumption of food. Humans and other vertebrates store glycogen in liver and muscle cells. But why do we make these? Who cares about sugars?
Well, let me tell you. Polysaccharides are used to store energy and structural materials. As a factory, we release glucose through hydrolysis of glycogen when our humans' demand for sugar increases. Hydrolysis is basically the opposite of dehydration synthesis. By adding water to a polymer, it breaks down into monomers. Now let's move on to the Nucleic Acid Station. Here we make DNA and RNA, or deoxyribonucleic acid and ribonucleic acid.
DNA provides a genetic material that organisms inherit from their parents. One molecule can carry several hundred or more genes.
This is adenine, thymine, cytosine, and guanine. DNA is in the shape of a double helix with base pairs that fit together in certain combinations. Adenine always pairs with thymine and guanine always pairs with cytosine. The base pairs are joined by hydrogen bonds. Red is adenine, green is thymine, orange is cytosine, yellow is guanine. Adenine, guanine, and cytosine can be found in DNA and RNA, but thymine is only found in DNA and uracil only in RNA.
DNA provides direct RNA synthesis and through RNA controls protein synthesis. RNA specifies a unique function in cells including enzyme catalysis.
Nucleic acids are micromolecules that exist as polymers called polynucleotides, so the monomers are just called nucleotides. A polynucleotide has a sugar phosphate backbone with various nitrogenous bases attached to it. Nucleic acids are formed by repeated dehydration synthesis reactions between nucleotides.
Water is a product in dehydration synthesis, while water is a reactant in hydrolysis, where polynucleotides are broken down into nucleotides.
Each nucleotide is composed of a nitrogenous base, a 5-carbon sugar called a pentose, and a phosphate group. Without the phosphate group, it would just be called a nucleoside. A nucleoside is made of a nitrogenous base and a sugar. There are two families of nitrogenous bases: pyrimides, which consists of cytosine, thymine, and uracil, and purines, which consists of adenine and guanine. The sugar that connects to the nitrogenous base is ribose for RNA and deoxyribose in DNA. They're basically the same, except that deoxyribose lacks an oxygen atom, hence the name deoxyribose.
Max!
What?
Can you show them the rest of the factory? I need a restroom break.
Okay. In this station, we make a special product called the lipids, a group of compounds that do not mix well with water. Lipids are special because they are not a true polymer and are not big enough to be a macromolecule. We make 3 types of lipids: phospholipids, fats, and steroids. Let's start with the fats. Fats hold the energy you need for a longer period of time. Fats consist of 2 types of molecules, glycerols and fatty acids. Fats consist of 3 fatty acids linked to one glycerol molecule called trisoglycerol. We connect 2 molecules by ester linkage, a bond between a hydroxyl group in the glycerol and the carboxyl group in a fatty acid. Through dehydration synthesis, we use our enzymes to bring these 2 molecules together in a time-efficient process that involves the loss of a water molecule from the reacting molecule. There are multiple types of fats, 2 of which are saturated and unsaturated fats. Unsaturated and saturated come from hydrocarbon chains of the fatty acids. Saturated fatty acids are literally saturated with hydrogen, meaning that the carbon skeleton is filled with hydrogen due to only single bonds in the chain. Unsaturated fatty acids, meanwhile, have double bonds caused by the removal of hydrogen atoms. Also, fatty acids can be broken into monomers by a hydrogenation reaction. Another lipid we make is called phospholipids. It is an essential material for cell membranes. Phospholipids consist of 2 fatty acids, glycerol, a phosphate group, and choline. Phospholipids consist of 2 fatty acids, glycerol, a phosphate group, and choline. Phosphate groups, glycerol, and choline form a hydrophilic head. Hydrophilic heads arrange itself to be on the outside of the membrane. Meanwhile, fatty acids form hydrophilic tails, which are always on the inside of the membrane and avoid water. Similar to fats, phospholipids are linked by ester linkages and can be broken down by hydrolysis. These are steroids, a type of lipid characterized by a carbon skeleton consisting of 4 rings with various chemical groups attached. Many hormones are steroids. One notable example of steroids is cholesterol. It is a common component of animal cell membranes and is also the precursor from which other steroids are synthesized. In this part of the factory, we make a new revolutionary product/polymer called protein, a new functional biological molecule consisting of 1 or more polypeptides folded and coiled into a specific 3-dimensional structure. It covers various functions including enzymes, defensive purposes, and storage. We make the proteins using amino acids, which are organic molecules that are monomers possessing both carboxyl and amino groups. This is an example of an amino acid. We connect the 2 chemical groups using peptide bonds, a bond created by a dehydration synthesis. The repeat of this process causes the formation of polypeptide, a polymer of amino acids. At one end of the polypeptide, there is a free amino group, also called amino end. On the opposite, there is a free carboxyl group. The repeating sequence of these atoms form peptide backbones, and anything that extends from the backbone are the side chains of amino acids. This is an example of a polypeptide. A protein's specific structure determines how it works. There are 4 levels of structures. Primary level is basically a unique sequence of amino acids that determine the function of proteins. Each one of the positions shown is occupied by a type of amino acid. They are arranged like arrangements of letters in a very long word. Primary structures are determined by inherited genetic information. The secondary level structure is a coil and a fold. They are the result of hydrogen bonds between the constituents of the polypeptide backbones. The model in blue is an alpha helix, which is a coil held together by hydrogen bonds every 4th amino acid, while the model in black are beta sheets, which are 2 or more regions of the polypeptide chains lying side by side, which are linked by hydrogen bonds between parts of the 2 parallel polypeptide backbones. The tertiary structure is the overall shape of a polypeptide resulting from interactions between side chains of various amino acids. One of the interactions are hydrophobic interactions, where the nonpolar side chains cluster in the middle of the protein to avoid water, thus resulting in the coil shape of the structure. Another interaction are hydrogen bonds, which are weak chemical bonds that are formed between a slightly positive hydrogen atom and slightly negative end of a molecule. Then there are ionic bonds, which are chemical bonds between 2 oppositely charged charged ions that help stabilize the tertiary structure. Then there are disulfide bridges, which are covalent bonds between two cysteine monomers formed from their side chains, amino acids and sulfur hydryl groups, and brought by the folding of the protein. Finally, there are van der Waals interactions, which are weak attractions between molecules or parts of molecules that result from accumulation of electrons in those parts. The hemoglobin is a quaternary structure. A quaternary structure is the overall protein structure that results from the bonding of polypeptide subunits. A protein needs to be in a right environment in order to be in its native shapes. If its physical or chemical environments are altered, then the protein would lose its native shapes in a change called denaturation and unravels into a biologically inactive protein. If the normal condition was restored, the protein can be changed back in a process called renaturation. One such example is the white part of the egg, where denaturation causes it to become a solid and insoluble. Proteins, like all polymers, can be broken down into monomers by hydrolysis. Now that we've reached our quota of macromolecules for the day, it's time to deliver it to all the cells in the body.
I feel tired. Can I have some carbs?
Yeah, sure, here.
About to duplicate. I also need some DNA for duplication.
Fine, here.
Oh yeah, can you give me some lipids and proteins?
Bruh. Fine, here you go.
Thank you. Bye.
See ya.